I can see it.
Okay, I'll just start it. Is that okay?
Yeah, please go ahead.
Okay. So first of all, I would like to apologize for the issue of internet connection for something we don't understand. It's not going well. And but today it's my great honor to share with you some of our recent research work on the neuroimmune regulations. And I would like to thank Dr. Wu and Dr. and Xiaolei for inviting me to give this talk to the community. And the title of my today's talk is about the neuroimmune regulation of spleen immunity and beyond. And as a very brief introduction of this field, as everyone here knows that our psychological status of a person could profoundly affect his or her health. For example, when people is under chronic stress, such as fear, anxiety, depression, or social defeat, often his or her health would deteriorate over time. On the other hand, when people is under relaxation, such as rest or placebo effect in the clinical trials, his or her health would improve over time. Therefore, such neuroimmune interactions has been generally recognized as a very important part of the regulation network in the body. And then, therefore, it's not surprising that the mechanism of neuroimmune interaction in the body has been subjected to intensive research in past decades. For example, in the past research, it's been documented that the classical hormonal mechanism is an important part of the neuroimmune regulation network in the body. And here just show some example of the classical hormonal regulation. And in one of the pathways, under the stress conditions, the brain will release a hormone called ACTH hormone. And this hormone into the blood circulation will then function into the adrenal gland to stimulate the secretion of the cortisol from the adrenal cortex. And then this cortisol in the circulation would function as a hormone to regulate the immune effect in the lymph node organs, such as spleen, lymph node, bone marrow, or other body tissues, such as lung, gut, or other vital organs, such as liver. And therefore, this hormonal regulation could exert a very broad immune regulation effect under stress conditions. In addition to the cortisol, the adrenal gland could also secrete another important hormone known as epinephrine under the stress conditions. And epinephrine in the blood circulation could also function as a hormone to regulate the immune functions in the various organs. However, in addition to these hormonal mechanisms, in the past 2 decades, it's become increasing evidence that in addition to the hormonal regulations through the blood circulations, the direct neuron input into the different organs or tissues could exert direct immune modulation functions through the release of different neurotransmitters, such as epinephrine, acetylcholine, or other neurotransmitters we could talk about later. And such direct immune modulation afforded by the neuron signals has become a new frontier of the research in neuroimmune regulation field. And that is the particular focus of my lab. And we would like to understand how the different immune organs, the classical immune organs, such as lymph node organs or the body tissues, could be regulated by the direct input, by the direct neural input within themselves. And therefore, today, I will just give you some of the examples of our research focusing on the spleen immunities. And I would like to talk about how we utilize some of the advanced imaging technique to analyze the neural input into the spleen or other lymph node organs in the different species. And then I would like to give you some examples as to how the different immune organs or the different neural input could exert the diversified immune regulation effect. And before I'm going to the details of my research about the neuroimmune regulation of the spleen immunity, I would like to give a perspective, give an overview of the imaging technique from a perspective of a neuroscientist. As a neuroscientist, we all know that is from the time of Cahal, people already utilize the 2D sections and together with the silver staining to reveal the complexity and beauty of neural systems. And over 1 century passed, and nowadays we have many advanced genetic labeling techniques and immune labeling techniques, also such as the brain bone mice shown here, and to see the different connections or the intrinsic complexity of the nervous systems. However, it's very important to realize that from the time of Cahal to nowadays, most of analysis from perspective of neuroscientists are based on the 2D sections. However, we all know that our nervous system is a 3D dimensional structures, and this 2D analysis would inevitably obscure our appreciation of the physiological or pathological structures of the nervous systems in the 3D dimension. And I really would like to use it as this artwork to elucidate this point. As you can see in this art exhibited in London about 10 years ago, exactly 10 years ago, you can see that is on every single sections, it will show only irregular structures. However, only upon we combine all the sections together, we could see this kind of the body structure of a human being. And therefore, to overcome this technical shortcoming based on the 2D sections, my lab has been particularly interested in utilizing the advanced 3D imaging technique to assess the neuroimmune interactions in different bodies, in different tissues or organs. Here is showing some of the highlights of the technical aspect of this 3D imaging technique. And first of all, we would like to utilizing the advanced imaging technique to do the immune labeling.
I think the speaker's internet dropped off again. Just give it a few minutes. Sorry about that.
I'll go ahead. I apologize again. It's very embarrassing, at least. And then I would say that is my lab has been particularly interested in to use the 3D imaging to assess the neuroimmune interaction in different organs. One particular aspect of the advantage of our technique is that we could perform the immune labeling of the intact S-section tissues. As you will see in the following examples, all the tissue imaging I show you are actually intact S-section tissues with the immune labeling technique. And then we'll perform the tissue clearing, tissue clearing to enable the removal of the light absorbing molecules as well as to match the refraction indexes. And therefore, the tissue become almost completely transparent, which enable us to do the light sheet imaging to assess the neuron structures or the immune structures in the intact tissues. And therefore, with that technique, we firstly would like to see how the neuron structure will be present in different mouse lymph node organs. We start from the mouse spleen. Here is showing an example of the intact S-section spleen of the adult mice. And the length of the spleen is over 1 centimeter and the width is about 5 millimeter. And then you can see that is after a procedure, the spleen become almost completely transparent. And then we can perform the immune labeling of the synapse, which show a pan-neural markers. And I will try to play the movie to see whether you can see it. And with that imaging technique, we could reveal for the first time in the field, there will be a very dense presence of the neural structures in the spleen. And moreover, we can see that these neural structures show a very organized flow-like structures throughout the entire parenchyma of the spleen. And then with that, we will see that is with the 3D imaging technique, we would able to reveal the very unique structure of the neural innervation in the spleen. And moreover, from the optical section enabled by the light sheet imaging, you could see that is the sympathetic, the neural innervations are present throughout the entire parenchyma of the spleen. Just to make sure what we really see here are neural innervations, we also use PGP 9.5, which is another pan-neural marker to perform the 3D imaging As you can see, the PGP 9 to the synapse phasing, just to confirm the dense presence of neural innervation in the adult mice spleen. And then we wanted to see how to confirm what type of the neural innervation are present in the mouse spleen. To this end, together with TH, tyrosine hydroxylase, which is a sympathetic marker. You could see that there's almost complete overlapping of the TH together with synapse phasing in this 3D imaging overlapping. Moreover, the TH whole tissue immune labeling gave to the same pattern of the synapse phasing immune labeling. Basically, there's a very organized flow-like structure of sympathetic innervation input in the spleen. To solve this issue, we first perform the whole tissue immunolabeling of Weichheit, which is a parasympathetic marker. You could see from this imaging, there's almost no detectable presence of parasympathetic innervations in the mouse spleens. Moreover, we cross the specific reporter mice, which is a check and then imaging the EGIP marker and you can see that is similar to what we observe with Weichheit immunolabeling. There's almost no detectable parasympathetic cholinergic parasympathetic input in the mouse spleens. Therefore, with this imaging technique, we are able to confirm first there will be very parasympathetic input. With this, like to examine some of the spatial distribution of the sympathetic innervation in the mouse spleen. And for example, here we perform the co-immunolabeling with TH, just to remind you, is a sympathetic marker together with CD169, which is a marginal zone macrophage marker. You can see that is the CD169 immunolabeling gave rise to this kind of follicle structures, basically making out the marginal zone structures throughout the spleen. Moreover, the co-immunolabeling of TH together with CD169 with the high magnification imaging clearly show that is the sympathetic innervations are present both inside as well as outside of the follicles. Moreover, the co-immunolabeling of TH together with PCAM1 is the vesicular endothelial marker, show that is most of the TH positive sympathetic fibers are actually spatially engaged with blood vessels within the spleen. With this analysis, we then answer the very important question is that because there's such an interesting structure of sympathetic innervations in the spleen, whether such structures would also be present in other mouse inferior organs. And for this purpose, we also do a systematic analysis of the sympathetic structures. We would like to see whether there will be any, we also want to examine other mouse lymph node organs. And we first do the bone marrow, as you see that is, here is a piece of the femur from the adult mice, and the length of the femur is over 1 centimeter, and the width of femur is about 5 millimeter. And again, with our 3D imaging technique, after the procedure, you can see the entire femur become almost completely transparent, which enable us to perform the 3D imaging analysis of the neural innervations within the bone marrow inside of femur. And you can see here, here's the movie that we perform the whole tissue immunolabeling of TH. Just to remind you, is the sympathetic markers. In the first part of the movie is showing the last sheet scanning of the entire femur of the bone marrow, and you can see that is because the tissue is completely transparent, the last sheet could penetrate through the entire tissue. And in the second part of the movie, there will be a 3D reconstruction of the entire sympathetic architecture in the bone marrow of the femur. And you can see that is in contrast to what we observe in the spleen, which show a very organized flower-like structures. The sympathetic innervation in the bone marrow of the femur show a kind of tree-like or branch-like structure, distinguish the innervation patterns of the bone marrow from the spleen. And moreover, we also performed analysis of other mouse lymph node organs, such as a lymph node. Here is another example of the whole tissue immunolabeling of the sympathetic fibers in the linguinal lymph node. And you can see that is with the last sheet scanning, it's very obvious that in contrast to what we observe in the spleen or the bone marrow, while there are sympathetic fibers present in the lymph node, in the mouse linguinal lymph node, they are actually exclusively present on the cell surface, on the tissue surface, but not inside of the parenchyma of the tissues. This result suggests a very interesting possibility that is, well, there will be sympathetic presence of the, there will be sympathetic presence in the different mouse inguinal lymph nodes, because they have different tissue localization structures, whether it's either is the cell surface localization or is in the parenchyma localization. They may exert different properties. Or moreover, which is just a summary of what we have done to analyze the sympathetic innervations in different mouse lymph node organs. And for example, here shown that is in the thymus of adult mice, there are almost no detectable, very few sympathetic fibers present in the mouse thymus. However, notably, I want to highlight that is there will be very strong Th positive T cells labeled in the thymus, which is consistent with the previous findings, suggest that certain T lymphocytes might be able to secrete catecholamines under certain conditions. And for other lymph node organs, such as the lymph node, subcutaneous lymph node, or the myocardial lymph node, you could see that is while the Th staining gave rise to the sympathetic innervations, such sympathetic innervations are kind of irregular shape. Moreover, such sympathetic innervations are exclusively present on the cell's tissue surface, but not inside of the parenchyma. And finally, it's very interesting to note that is in the pear patch, which is another very important mouse lymph node organs in the gut, there will be very interesting mesh-like structures of Th positive sympathetic innervations. However, even though there will be very dense sympathetic presence in the mouse pear patches, the optical section of light sheet imaging clearly reveal that is those sympathetic fibers are only present in the tissue surface, but not inside the parenchymas. In contrast, there will be almost no presence of VCHAD positive parasympathetic innervations in the thymus, subcutaneous lymph node, or metatarsial lymph node. Again, while there will be some of the cholinergic fibers in the pear patches, these cholinergic fibers are only present on the tissue surface. Therefore, taken together, those results have demonstrated that in the mouse lymph node organs, there will be different patterns of sympathetic innervations in different organs. And in particular, the spleen have a very dense flower-like structures within the parenchyma. And with that, we want to ask ourselves an important question is that because we perform the analysis only currently in the mice, whether such different distribution of the neuroinnervations would be conserved in other mammals, in particular in primates. And for that purpose, we went on our analysis to the non-human primates, basically the macaque monkey, to see whether we observe in the mouse would be conserved in the monkey. And here we harvest the spleen from the adult monkey, and then so you can see that is here we harvest the monkey spleen, and with our procedure, the spleen become almost completely transparent. And moreover, we perform the whole tissue immune labeling of PGP 9.5. To remind you, is the pan-neural marker. You can see that is the 3D imaging reveal very dense and flower-like structures of neuroinnervation in the monkey spleen. And moreover, we also perform the Th whole tissue immune labeling, which is a sympathetic marker. Here you can see that is similar to what we observe in the mouse monkey. There is a very dense presence of sympathetic structures within the monkey spleen also. And very important to note that is, in contrast to the presence of sympathetic input to the monkey spleen, there will be almost no detectable presence of VCHAD positive parasympathetic fibers in the monkey spleen, which substantiates the conclusion that is the spleen is exclusively innervated by the sympathetic but not parasympathetic input. And moreover, we also carry out this analysis of the monkey lymph node organs. And just to cut story short, the conclusion is very similar to what we observe in the mouse spleen or the mouse lymph node. That is, in the monkey lymph node, while there's some irregular presence of the neuroinnervations or sympathetic innervations, those innervations are exclusively present on the lymph node tissue surface, but not inside of its parenchyma. And finally, we also would like to see how it looks like in the monkey pear patches. The monkey pear patches actually gave rise to similar mesh-like neuron structures, similar to that in the mouse pear patches. And but also, it's very important to note as either for the PGP 9.5 positive labeling to show the total neuroinnervation, or the Th labeling to see the sympathetic innervation, or the VCHAD to see the parasympathetic innervations. Those innervations in the monkey pear patches are only present in the tissue surface, but not inside the parenchyma. Basically, there's a black hole here. You can clearly see there's no neuroinnervation at all. Therefore, this result confirm what we observe in the mouse, but also very importantly to show that is even in the non-human primates lymph node organs, the neural innervation patterns are dramatically different from different organs, and the spleen is only tissues that contain the dense sympathetic innervation fibers in the parenchyma. And finally, we extend this analysis and the conclusion to the human samples. In collaboration with Peking University's 3rd hospitals, we are able to harvest the human spleen from the adult patients. And then we perform the TH whole tissue immune labeling, and similar to what you'll see in the mouse or the monkey spleen, there will be a very dense presence of sympathetic innervation in the human spleen. But there will be almost no presence or only a few fiber of the VCHAT positive parasympathetic innervations. We believe that this result, those results garnered from the mice, non-human primate, and the human spleen has helped to settle down the controversy in the field. That is, there will be only sympathetic but not parasympathetic input to the mammalian spleens. And therefore, even if the parasympathetic might exert any immune regulation functions in the spleen immunity, it could not exert through direct interactions. And we also do the same analysis for the human lymph node organs, such as the human lymph node or the human pair patches. The conclusion is also the same. That is, for the lymph node organs, such as human lymph node and pair patches, well, there will be the neural innervation present. Those neural innervations are present exclusively on the tissue surface, but not inside of the parenchyma, which suggests that those neural innervations will not exert or not able to exert the direct regulation of the immune response happening inside the parenchymas of this immune organ. I will just give you some example to show that is aimed by the advanced 3D imaging techniques. We are able to systematically examine the neural innervation patterns in the mouse, non-human primate, and human lymph node organs. And among all the lymph node organs we examined, it's become very clear that is the spleen has a very dense sympathetic innervations that are uniquely present in its parenchyma. In contrast, other lymph node organs, such as lymph node or the pair patches, will have some neural innervations on the tissue surface, will not have any neural innervations in the parenchymas. And also, it's very important to emphasize that the spleen of different species only contain the sympathetic but not parasympathetic innervations, which help to settle down the controversies present in the field. And with that findings, we would like to went on to answer the question or ask the question: if there will be such unique dense presence of sympathetic innervations in the different species of the spleen, what's the potential function of such sympathetic innervations? And to answer the questions, I will give you some examples and to show you some technique or the mouse models we are going to use to address these very important questions. In the first part of this functional analysis, I would like to introduce a very useful genetic models that could specifically ablate the sympathetic innervations in the mouse spleen. Here you can see that is we crossed a specific genetic model, which is TRK conditional allele. For information, TRK is a specific receptor for the neural growth factor, also known as NGF. NGF and the TRK signal has been shown or known to be very essential for the sympathetic development in the mammals. Therefore, by cross the TRK conditional allele, basically specific knockout the TRK receptor in the sympathetic populations, we are able to block the development of the sympathetic networks throughout the body during development. And you can see that is this TRK mice, the spleen of the TRK mice look almost same as the control mice, which is TRK white type allele. And however, when we perform the 3D imaging analysis with TH, which is sympathetic markers, you could clearly see that is compared to the very dense presence of sympathetic innervations in the white type control mice, the TRK conditional knockout mice almost completely lost their sympathetic input in their spleen. And moreover, when we perform the whole tissue analysis of the synaptophysin, which is pan-neural marker, it also gave rise to almost same conclusion, that is the total neural innervations are also completed, almost completed question how such loss of sympathetic signals would cause any effect in the perspective of immunology. And the first thing we would like to examine is that if we can use this genetic model to remove the sympathetic fibers in the mouse spleens, whether it will affect some of the general immune populations in the mouse spleen. But the answer is no. Here showing that some of the example of the facts analysis compared to the wild-type spleen or the TRK spleen. As you can see from this very simple analysis, the major immune cell populations such as B lymphocyte, CD4 lymphocyte, CD4 T lymphocyte, CD8 T lymphocyte, or dendritic cell or macrophages are generally speaking normal compared to two different conditions. Moreover, the whole tissue immune labeling of CD169, as you may remember, is a marker for the marginal zone macrophages, also show the similar structure of the follicle structures compared to the wild-type or the TRK knockout mice. Therefore, this result suggests that the loss of sympathetic input would generally does not affect the major immune cell population in the spleen. And then the question is that if the sympathetic loss will not cause a general loss or change of the major cell population, immune cell population in the spleen, what the function of the sympathetic innervation in the spleen would be. And we test several models that could strongly stimulate the innate immune response in the spleen, such as the antivirus innate immunity or the antibacterial innate immunity. We particularly found out that the loss of the sympathetic input could enhance or significantly enhance the antibacterial innate immunity in the mouse spleen. For example, we could treat mice with LPS from gram-negative bacteria to elicit antibacterial innate immunity in the spleen. Cell analysis of the mRNA levels of different cytokines or chemokines in the spleen. The treatment of the LPS could cause a significant upregulation of different cytokines or chemokines in the wild-type spleens. But more importantly, in the TRK knockout mice, which lost their sympathetic input in their spleen, such treatment of LPS would cause a significantly increased expression of the TNF-alpha, interferon beta, interferon 6, or other cytokine or chemokines. This result clearly shows that is the loss of sympathetic response. Moreover, the recruitment response, the recruitment of all mice. And moreover, such an enhancement of the antibacterial innate immunity could be translated into a protective effect against the bacterial infection. Here, we inoculate the TRK knockout mice or the wild-type litter mice with the gram-negative bacteria. As you can see, about 4 hours after the bacterial inoculation, while there will be significant presence of the bacteria still in the spleen, there will be almost no presence of the bacteria at all in the spleen, suggesting that such enhanced antibacterial innate immune response could be translated to a more effective clearance of the bacterial infection. Moreover, when we examine the survival curve of the TRK knockout mice compared to the THK wild-type mice, you can see that is such inoculation of the lethal dose of bacteria would eliminate or would cause the death over 80% of the wild-type mice, but over 60% of the TRK knockout mice would survive this bacterial challenge. Therefore, this result gave to the conclusion that is the loss of sympathetic input could help to promote the antibacterial immunity in the spleen. And then what's the potential molecular mechanism have been also a debated issue as to what is really the neurotransmitter that regulates the neuroimmune regulation in the spleen. Well, some people propose that the norepinephrine could be the direct regulation, could exert its direct regulation. There will be also reports or indicate that the acetylcholine is another important neurotransmitter, may also have the effect on the immune function of the spleen. To address this issue, we just use the very simple in vitro culture systems. It's in the in vitro culture systems of the bone marrow-derived macrophages to mimic the antibacterial innate immune response. We could see that is in the wild-type the bone marrow-derived macrophages, the treatment of sympathetic neurotransmitter norepinephrine would directly decrease the expression of different cytokines and chemokines that can be enhanced, that can be otherwise enhanced by the LPS treatment. This result suggests that the immune regulations exerted by the sympathetic input could, or at least could, be contributed to the norepinephrine effect. Moreover, such norepinephrine regulation of the innate immune response could be strongly or completely blocked by the ADRB2 knockout mice, suggests that this effect, such as, for example, in the wild-type mice, the norepinephrine treatment would significantly decrease the expression of TNF-alpha or interleukin-6. However, such inhibitory effect of the norepinephrine would be completely reverted in the bone marrow-derived macrophages of ADRB2 knockout mice. Those results suggest that the norepinephrine would exert its immunomodulatory effect mainly through the ADRB2 receptor. And for the second part, I would like to give a very brief overview of our analysis through the genetic model to see how the sympathetic input in the spleen may exert a very important role in the antibacterial innate immunity. However, a very important question come out from this result is that if the sympathetic input in the spleen would function to suppress antibacterial innate immunity, what's the evolutionary benefit, or generally speaking, what's the benefit of such a suppressive effect of sympathetic signals? Because if we could release such sympathetic input suppression, in theory, we could enhance the general health of the animal or even human beings against bacterial infections. So to answer this very important question as to what's the functional relevance or the significance of such suppressive effect of spleen antibacterial innate immunity, I would like to introduce a second model we utilize in the mice to examine the particular function of a sympathetic input in the mouse spleen. That is, we could use the surgical ablation to completely remove the sympathetic signals in the mouse spleen. Here is showing that is compared to the mouse surgery, the sympathectomy, basically the surgical procedure, could completely remove the Th positive sympathetic fibers in the mouse spleens. Again, similar to what we observed in the TK knockout mice spleen, such loss of the sympathetic input in the spleen by surgical approach does not affect the general presence of major immune cell population in the spleen, nor the follicle structure present in the spleen as shown by the whole tissue immune labeling of CD169. And then we would like to see if this sympathetic loss this time could be very specific to the spleen, would there any detrimental effect, or in addition to its protective effect, whether there will be any detrimental effect caused by such sympathetic loss. And we do find out there will be a detrimental effect. And here, I would like to introduce to you, as everyone here knows, is a very important anti-inflammatory factor called interleukin 10. Interleukin 10 is probably one of the major central immunosuppressive factors in our bodies. And it's very important for the interleukin 10 to exert the immunosuppressive effect to maintain the immune homeostasis in the body. And we already know that is upon the LPS treatment, in addition to the expression of all the pro-inflammatory cytokines or chemokines in the spleen, there will be also a significant upregulation as shown in the blue bar in the mouse condition, upregulation of interleukin 10 production in the spleen. This interleukin production could be potentially important to protect against the sepsis shock in the mice or in the human body, in the human bodies. However, we found that upon the sympathetic loss in the spleen, such interleukin 10 expression got significantly suppressed. And notably, this effect is very tissue specific because our sympathectomy procedure only affects the spleen sympathetic innervations. It did not touch the liver or inguinal lymph node. The interleukin 10 expression in the liver or inguinal lymph node are not affected as expected. However, just to show the importance of the spleen-derived interleukin 10, such loss of interleukin 10 expression in the spleen would be sufficient to cause a decrease of the plasma level of interleukin 10 proteins in the animal. And moreover, such loss or decreased level of interleukin enhance a significant worsened death in the septic models. As you can see, that is upon the LPS induced in the toxemia, basically a model for sepsis, in the normal conditions, about 20% of animals die. However, upon loss of their sympathetic innervation in the spleen, over 70% of animals will die to the septic conditions. And those deaths could be completely reverted if we could treat animal with recombinant interleukin 10 protein. And in addition to the LPS-induced septic conditions, we also examine the potential effect on the CLP, basically cycle ligation and punctuation, in short, called CIP model of sepsis. And similar to what we observed in the LPS treatment conditions, while in the control conditions, there will be a significant increase of the interleukin production in the spleen. The loss of the sympathetic innervations in the spleen causes a dramatic decrease of interleukin productions in the spleen, which could be translated into a decreased level of interleukin proteins in the plasma. And again, just to show the importance of such interleukin response against sepsis, while in the control conditions, about 50% of the mice would survive the CLP treatment or the procedure. Over 90% of the mice will die in the sepsis models. And moreover, such susceptibility to the sepsis conditions could be reverted completely, almost completely, by the treatment of interleukin proteins. Therefore, this result reveals a very interesting aspect of sympathetic regulation of spleen immunity. That is, well, the sympathetic signals could exert suppression effect against the antibacterial innate immune response. It's also important for the sympathetic signal to promote the interleukin response to protect against sepsis. And just to confirm this conclusion, that is, the local sympathetic signals is indeed essential for the spleen production of interleukin upon the sepsis conditions. I would like to rule out, or we would like to rule out, the potential involvement of the classical hormonal mechanisms. As just to remind you, in the introduction part, we talk about how the adrenal gland could secrete the cortisol or epinephrine, which may potentially regulate the immune response in different organs, including the spleen. And to rule out the potential involvement of adrenal gland-derived signals, we perform the bilateral adrenalectomy. Basically, we surgically remove both of the adrenal glands from the mice, and then subject the mice to the LPS challenge. And you could see that is, compared to the mouse conditions, the loss of the adrenal gland does not affect the TNF-alpha interferon beta, such those pro-inflammatory factors expression in the spleen upon the LPS treatment. Moreover, the LPS-induced interchain expression are also not affected without the adrenal gland-derived signals. Furthermore, the survival rate of the mice upon the LPS-induced sepsis conditions also are comparable with the mouse conditions. All these results suggest that the adrenal gland-derived signals may not exert a major role in this context of the spleen immunity. And finally, we would like to confirm or substantiate our conclusion, that is, the sympathetic input to the spleen would be sufficient to indicate, sufficient to instruct the spleen response of the interleukin. And to this end, we use the optogenetic approach. Here, we use the intrasplenic injection of AAV virus. And here you can see that this AAV virus express a reporter EGIP in a crate-dependent manners. And therefore, we inject this AAV virus in the TH crate mice. Basically, we use the crate, TH crate of sympathetic expression to cause expression of the EGIP. The whole tissue immunolabeling of the EGIP show that this AAV labeling is very efficient, because about over 70% or close to 80% of the TH positive sympathetic fibers could be labeled by such intrasplenic injection of AAV. And moreover, this AAV treatment or labeling is also tissue specific, because in the same mice, if we took the adrenal gland, liver, or inguinal lymph node, perform the whole tissue analysis for the EGIP expression together with TH analysis for the sympathetic marker, you could see that there are almost no detectable presence of EGIP in the different tissues, suggesting that AAV injection is relatively spleen specific. And then we ask a question: if we could use the chemogenetic approach to directly control, to inhibit or enhance the sympathetic input in the spleen, what the result would be in terms of the interleukin response. And here you can see that we first utilize a chemogenetic receptor that it could be an inhibitor receptor upon the CNO treatment. We could see that is upon the signal induce the chemogenetic inhibition of the sympathetic input to the spleen, there will be a significant decrease of interleukin-10 production in the spleen upon the LPS treatment, but not in other tissues such as liver or inguinal lymph node. Moreover, such decrease of spleen interleukin-10 production could be translated to a decreased level of plasma interleukin protein. In contrast, or conversely, if we utilize a chemogenetic activator in the CNO treatment, we can see that is upon the LPS treatment, the interleukin production by the spleen will be significantly enhanced upon the LPS treatment. And such enhanced interleukin-10 production in the spleen could also be translated to enhance the interleukin-10 production in the plasma. Therefore, this result confirmed the very important conclusion that is the sympathetic input or signal is sufficient to designate the interleukin-10 production. And finally, we would like to see that is whether such regulation could be a direct role of sympathetic signal in terms of the norepinephrine neurotransmitter. And we first analyzed the interleukin-10 positive immune cells together with the Th fibers in the spleen upon the LPS treatment with the 3D imaging. And those 3D imaging gave us to a very striking pattern that you can see that is, well, there's many interleukin-10 positive immune cells in the spleen. Those interleukin-10 positive immune cells tend to cluster around or in adjacent to the sympathetic fibers. Those results gave a very convincing evidence suggest that the sympathetic signal would have a spatial engagement of interleukin production in the spleen. And in support of this notion, we found that in the in vitro cultured mouse splenocyte, the norepinephrine, which is a neurotransmitter for the sympathetic signal, would be sufficient to increase the splenocyte production of interleukin-10, and such result would be completely abolished in the splenocyte from ADRB2 knockout mice. Moreover, we also test this effect in the human splenocyte. You could see that is, while the human splenocyte treated by LPS alone only gave a relatively minor increase of interleukin production, the splenocyte treated with LPS together with ADRB2 agonist would cause a dramatic increase of interleukin production in the human splenocyte. And finally, we are able to confirm that is this ADRB2 agonist, or generally speaking, the ADRB2 signal could effectively revert the sepsis susceptibility caused by the sympathetic loss. Basically, if we have the mice subject to the spleen sympathectomy, basically remove their sympathetic signal, but then give their IV treatment of the formoterol, which is ADRB2 agonist, you could see this ADRB2 agonist could significantly increase interleukin production in the spleen, as well as the interleukin present in the plasma. And finally, this ADRB2 agonist treatment would almost completely suppress the death caused by the LPS induced endotoxemia. So taking everything together, I talk with you. I give you some example as to how we utilize the 3D imaging technique to systematically examine the sympathetic or neural innervation patterns in the different lymph node organs of the mice, non-human primate, and human. Moreover, I give you an example to show that we can use the genetic model, the surgical model, or the optogenetic models, the chemogenetic models to control the sympathetic signals in the spleen. And by using these different models and disease models combined together, we're able to show that the local sympathetic signals through its neurotransmitter norepinephrine could, on the one hand, suppress the antibacterial innate immunity, basically suppress the production of pro-inflammatory cytokines such as TNF-alpha, interleukin-6, but also at the same time, on the other hand, could promote the production of interleukin-10 in the spleen. This bidirectional, this kind of bidirectional regulation of the spleen immunity by the sympathetic signals would give a very good example of how the local neural input in the different tissues could designate the tissue immunity under different contexts. Of course, as I just told you in the introduction, our lab has also been interested in other immune organs or other biotissues. And given the different tissues have different patterns of neural innervations, as you could expect or guess, that is, the different tissue immunity, the different tissues could have different mechanism of neuroimmune regulation. And finally, I would like to thank all the findings of my research. My findings the finding in my lab come from the CLS for attention. And finally, I will be received. Thank you.
Okay, so thank you so much, Jing. I think you can stay put like this because I just don't want to the video get disrupted. I think I think we can start the Q&A session. I think a lot of people already start to ask questions. First question actually I think is from Dan Yang. Dan Yang, do you want to go ahead to ask question? Dan Yang, are you there? Oh, okay. So I'm going to read her question. I think her question is whether this enhanced innate immune response is upon loss of sympathetic input specific to the spleen.
That is actually because our is not only affect the sympathetic input to the spleen, it also affect the sympathetic input organs such as the skin or the lung. We actually got the same conclusion is that the tissues, but in the